So I'm going to talk about network analysis, but in fact, I want to talk mostly about choice, making choice. So what we investigate, right? Which genes in this case. And this is, okay. This wasn't on. Okay. Yeah, thanks. This is a paper that we recently published on the topic of doing network analysis in the Atlantic salmon louse to identify important genes and modules of genes in the molting or developmental process. And this is mostly work of my PhD student, Jonhard, first author of this paper. So if you're interested in the nitty-gritty details, I need to refer to the method section or supplementary. But first, let's talk about motivation, because that is not quite clear. In the paper, it looks just we wanted to do network analysis, but we wanted to take a step back again, and we wanted to talk about priorities or prioritization, which is sort of glorified for ranking things by importance. So we often make decisions on importance, right, in our personal life, what to spend our money on, or we have these decisions made for us, for example, by Google. So, I mean, you get the Google results, and I guess a lot of us will not go to the second page and just click on the first results, relatively often at least. So, maybe we all agree at least that clicking through all the 60,000 results I got is not a good strategy for this. So we need to prioritize, especially when resources are scarce. Examples are very different. For example, medical triage has been a topic maybe discussed in a pandemic. And then, well, the Google ranking algorithms. We trust these because Google wouldn't tell us how they rank their pages. That's their trade secrets. In the biomedical context, however, we also have often too many genes, and in diseases mainly, we need like disease prioritization or gene prioritization for ranking disease genes, so which to investigate, and to identify genes that are likely associated with a disease. So there are different methods to do this. In the salmon louse, well, I talked about the genome. Now the only thing you need to know is the 14,000 genes, right? And they have been published, and even more non-coding genes. So what we can do and what we've seen a lot results already is gene knockdown by RNA interference or by RNA-mediated knockdown. Well, whatever. So this was published by Fire already in 1998. They did on C. elegans. And it has been successfully applied on the salmon louse since 2009. And the principle is simply we need, of course, controls and a knockdown. The double-stranded RNA is injected into the knockdown lice. They put on the fish. Then they are allowed to complete their life cycle and meticulously observed for 10 to maybe 70 days, and then a lot of sampling, photography, analysis is done, and we hope to see a change in phenotype, like with a heme scavenger receptor when it's knocked out, and then maybe the results end up in lice space, right? So, of course, there's some obstacles on the way. Not always does this work. This digestion of the mRNA by the RIS complex does not always yield a total reduction of protein, okay? But anyway, I mean, so it's possible, so we can knock down any gene in the genome, but we cannot knock down all genes in the genome. So I was trying to estimate what the global capacity of gene knockdowns is, in fact. So, well, in the SLC, over 8 years, we did about 400 experiments, maybe more. They ended up in lice space, so that's the ones I know of. So that's like 50, so be optimistic. So let's say the global capacity maybe is 100 gene knockdowns meticulously followed up over 10 to 70 days, right? Okay, how long does this take? Well, it's a very simple calculus. well, 140 years. That is not a very good ETA for what we need, so it is principally infeasible to do this. Even with other technologies, this would not change. If you had like CRISPR-Cas or something, it wouldn't change because you still need to follow up, right? You can do it on nuclei, but in principle, the nuclei experiment belongs to the cycle, right? Belongs to the life cycle. So in principle, you want to follow up through the whole life cycle. So genome-wide knockdown is not feasible in this organism, in parasites, right? So then if we cannot choose all genes, what's the probability of that we choose any gene that we see something, okay? So taking lysebase, okay, there are experiments there, and the phenotype is annotated. So here you see the phenotypes, the essentiality, and the phenotypic rates of, well, these 400 experiments in the pie chart. So this is no visible phenotype, and this is NA, I assume. This is also, I tried to follow it up, so this really means there is nothing to see here. Of course, I cannot exclude that there are some experiments who were not reported, right? But we really took care of trying to get everything in, really, except maybe some really completely failed attempts. So and then the first largest change in phenotype has to do with female reproduction. That's also clear because most is done on adult females, right? Most experiments. And then you get all the other phenotypes. So the phenotype success rate is approximately 25%, right? That's already quite good. In nuclei, it's only about 6%, seemingly, for the bath treatment. But that's still quite good, right? Because when you look at like human genes, for example, 9 to 10% are believed to be essential. And there was a big genome-wide RNA screen in Drosophila. In culture cell, they found only 2.2% essential double-stranded RNA that really showed a change in phenotype. So, I mean, this is sort of done by the outstanding expert in the field, so the choice should be great, right? So, but then the question is, okay, if we can't do everything, can the machine do better, right? Or an algorithm, that's what the bioinformatician would ask. Can we choose candidates with higher probability of success? So, and then, of course, we came to networks, because network analysis has been applied for choosing genes, candidate genes. Oh, sorry, I exited. Okay. Could somebody put it's a dangerous button. Could you put the presentation in presentation mode again, please? Yeah. Okay, but, well, networks, I just continue. Networks are, in principle, graphs, right? Graphs consist of vertices or nodes and edges, and then when they are graphs, we can use graph theory to analyze them. And they can be used for unsupervised learning, right? For example, for, graph clustering, okay? There are different kinds of networks, very different kinds, like computer networks, obviously. These are different topologies of computer networks, in fact. Some of these may be found in biological networks as well. The social networks, right? Protein-protein interaction networks are very well used for some organisms where you have such data. And then there are gene regulatory networks. They are directed graphs. There are gene co-expression networks, and they're based on correlations of gene expression. So some basic network terms I should repeat in this context, and some very basic properties, I think. I mean, it's almost as common as hierarchical clustering now, but still. So basic network properties is one, node centrality. So here's, right, here are the nodes. And we already see this node is central, in a sense. It has a lot of connections, and in particular, a lot of paths between nodes are going through it. That's between the centrality. And centrality, this is centrality lethality rule, CLR, that sort of determines the more central a node is, the higher the likelihood that its deletion be, well, lethal. I mean, it's sort of understandable. There might be a difference if you remove my profile or Mark Zuckerberg's profile from Facebook, but debatable. Anyway, modules, then the second term. Modules are sort of clusters of tightly connected nodes, so we have drawn these, they're tightly connected. And then when you have clusters or modules, guilt by association rule may apply. That means like if, for example, all my friends are crooks, maybe so am I. Of course, like in real life, this is even more debated than the first point. But in general, it's a helpful principle, and we will see that it can occur, but doesn't always occur, and it's not the solution for everything either. But yeah, that we knew maybe. So we needed training data. We needed a large data set, right, to train this, e.g. a time series to calculate gene correlations. And there are not so many large time series for salmonis, especially on gene expression. So we choose one on molting and development. Now, I need to explain another concept. That's molting and development, but only very briefly. So molting, the shedding and replacement of the cuticle and chitinous exoskeleton is common to ecdysozoa. It's regulated, among other things, by molting hormones at 20E. Their corresponding nuclear receptors, ECR, RXR, FTZ, they have been studied here. Also, this has been very well studied in Drosophila. It's an important cyclic developmental process. And here you see the pathway for making this molting hormone. And there are these so-called Halloween genes, spook and phantom, and these others involved in this pathway. So they make 28, or insect insect hormone biosynthesis. So we had this large instar-staged developmental time series from before. That was done by Christiane, Frank, and me. And yeah, we already saw there could be patterns discovered, and it needs to be well staged because growth also occurs throughout the instars. And also this is not super synchronized. So when you sample just by days, you could get very different lice within the instar. Some that are very close to molting, and some that are just molted. Also, so it's not good to sample by days, but then this this technique by staging them by cephalothorax length, by total length had been developed before also. And there's a big data matrix, and you see like, I think it's 66 samples, which is quite good. So it's mostly still the largest, well, should be careful with all the superlatives. Anyway. Sample grouping then, so we didn't take the young ones, we just took the middle instars and the old molting instars, so those that were close to molt or molting, into our analysis. And then we created gene co-expression networks, 3 of them. Old molting network, 27 samples, middle network had 27 samples, and the global network, we took all the samples except the young ones, and that is 45 samples. And then we identified, we built the network, and we identified modules using the common R package for this task. Also, we wanted to include something new, so include something we call orthologous phenotypes. So we need another organism where this whole genome screening is possible, okay? So we use that. There's a database called Genome RNA-I, and there's a lot of information in there, mostly loss of function. Phenotypes are there, and we found 3,059 salmon louse transcripts that had orthologs in a lot of loss of function phenotypes. So the workflow was like this. We had the gene expression matrix normalized already from the other paper. We calculate pairwise correlation, and you have to estimate scale-free parameters, calculate another matrix. So it's just a series of transformations until here. Then you use extrinsic and intrinsic data after you found the modules. And then we said, okay, there are modules. We need to identify the important modules first, okay? Because, I mean, there are lots of hubs maybe in these networks, but there are also lots of modules. So which module is important? That was our idea. So identify important modules and then identify the hubs in these important modules. And yeah, we use different centrality measures. I don't have time to go into detail here. We use 3. We call an absolute hub a hub that is sort of ranked 1 in all 3 centrality indices. And these are quite commonly used. Okay? So then, how did we choose modules? So we had like 3 different rules. First, we used non-preserved modules. We wanted to have modules that were specifically only there in molting. And then we wanted to have modules where the eigengene, like you can imagine that's like a centroid of the module, is similar to the molting process. So it varies with or correlated with the molting process. Or also we wanted modules that were significantly enriched in lethal or any orthologous phenotype in Drosophila. So which modules did we choose? We didn't have enough resources to evaluate all 8, so we choose 3, the best ones for each criteria. Here's the yellow-green unpreserved molting model. Violet is chosen for being enriched in phenotypes, and steel blue was selected for strong correlation with the phenotype, with molting. So how did they look? This is the eigengene vector. So you see they look like clusters, also these modules. And the eigengene, so here are these 3 modules. This one is the global one, is strongly correlated with the molting phenotype. You can see that the molting is differently regulated. Okay, then we have these. So we start with the yellow-green, was unpreserved. And yeah, what did we do here? We choose, of course, the top candidates from each of the modules, right? And this is to knock down. This is one, Ras-related protein ORAP1 was the annotation in Lice base. Then we choose this from the second module. And from the third, well, this was full of cuticle proteins. This is the guilt by association I talked about. Also, this one is the spook Halloween gene. Okay, so, but here we had a lot of cuticle proteins. So it was a bit boring, but there was one that wasn't annotated, so we took that. And, well, we took these. We additionally also used some. We took some that were more down the list. So we didn't only choose the top candidates to see how far down we could go. So we had, in total, we had 5 candidates to knock out and, experimentally validate. And there was this Ras one, the digestive organ expansion factor, this gene, ER membrane protein complex construct domain, and a hypothetical protein, the one I showed before in this. And did it work? Yes. So fortunately, yes. And, so 4 out of these 5 showed really stronger phenotypes. They were knocked down. And this one first delivered no X strings when it was done in adults, and when it was done in nauplii, then they did never transition from copperpot to Charlimus 1 on the fish, so we never found them. And then the others had, this one has also no X strings. This was further down the list. I think this was rank 10 or so. So it still worked somehow. And this one, they were just shorter, so this was a bit weaker, I would say. This one, well, there was something in the cephalothorax and stuff, but well, I don't really know about this. So I don't have time to list all of them. So we'll just mention 2, right? This one, this Ras-related protein, WARAP1, is the best blasted of this. It belongs to the family of small RAP GTPases. It is super highly conserved. It has 87% sequence identity to human RAP1A, but there's no good Drosophila autolog. That was maybe why we didn't see it immediately as a lethal phenotype, okay? So GTPases are general versatile molecular switches, and they regulate a bunch of different processes, mostly involved in trafficking between membranes, right? From the ER to the Golgi, and they are involved in integrin recycling, autophagosome formation. Yeah, they interact with the TOR pathway, and they are a human oncogene. So I found this nice site just to validate this. We didn't include this data, but RAP1A is essential in human experiments. is strongly selective and common essential. That's a very interesting side, in fact. So then the second one was a hypothetical protein, right, with the many cuticle-related genes and the spook gene on the module, possibly a cuticle-related protein, and we discovered an ortholog in Tigriopus, which has a domain, a chitin binding domain, and this might be, in fact, also a cuticle-related protein as well. So we can sort of restitute the thing, like the annotation. So the question is now, if this was successful, even with a small set, will the machines take over? This is maybe the wrong question because they already have, right? So computer algorithms and machine learning are becoming pervasive in decision making, and we propose that that should also be the case in sea lice biology because we have the tools now. Still, there's a lot to be improved, of course. So it has the potential to increase the probability of success in functional studies. And I mean, it's really interesting to just reflect, how do you choose the genes you're interested in and you're working with? And maybe, how do you think, will you still do it in the same way in the future, right? How could this be a valuable contribution to selecting genes with good probability of success? So in the end, I just want to conclude, well, there is a key, we found some, and this method somehow works, but of course we could only evaluate 5 candidates, but we hope that the community might pick the rest up because they're open, so just go ahead, take the genes if you want high probability of success, possibly. Thank you.